Hey everybody, so today we're going to talk about environmental effects on phenotype and chromosomal inheritance. Okay, so the environment can affect a gene's expression, meaning it can affect how that gene, what proteins that gene produces. And so if the environmental conditions change, the expression of the gene can change. And that's known as phenotypic plasticity. And so that's when one genotype can produce more than one phenotype. So an example is hydrangea plants. The color of the flower is based on the pH of the soil. So if you have acidic soil or like a low pH, like 2 or 3, you're going to get blue and lavender flowers. Whereas if you have basic soil, like 8 or 9 or whatever, then you'll get pink and red flowers. So you can kind of see the different flower colors down below. The other thing that's interesting about how the environment can change gene expression with phenotypic plasticity is the stomate density. So stomates, if you think of a leaf, stomates are the little holes that are on the underside of a leaf that allow carbon dioxide in and oxygen out. And so if you have low amounts of carbon dioxide in the air, then that's going to lead to more stomates being formed so they can get that carbon dioxide in. And if you have high amounts of carbon dioxide, then that will lead to fewer stomates being made. And another thing is that organisms can have the same genes but show different forms based on the environment. So a good example of this is if you look at identical twins when they're older, right? They look slightly different, and as they age, they look more and more different because they're in different environments. When they're growing up, they're eating the same food, they're having the same interactions, but as they get older and they become adults, they have different lifestyles. And so this is just a fun picture, and you can kind of see they look very, very similar, but they're not, completely identical. Okay, so we're going to talk about genetic disorders first and the different types of inheritance. So you're probably going to want to draw the pedigree and then write down these patterns. Okay, so just to review on pedigrees, males are squares and females are circles. And if you're shaded in, then that means you have whatever the trait is you're talking about. For this one, it's recessive. So remember, recessive, everybody who's shaded in would be little r, and everybody who's not shaded in would have at least one big R, and then, depending, like these two parents right here would both have to be big R, little r, so they could both give their little r to their son. So in recessive traits, this is usually skip generations because they have what's called carriers. And a carrier would be like these two parents. They're both heterozygous, and they're called a carrier because they carry that little r, they carry that recessive trait, but they don't show the phenotype because it's recessive, and that dominant one's going to cover over it. And the autosomal part means that it's on chromosome 1 to 22, so there's going to be about equal male and female incidence of who gets it. An example of this is sickle cell anemia, where your blood sickles, and then it can't carry oxygen properly to your organs. And I know we talked about that in biology before, and we'll talk about it more too. Okay, so the next one is a genetic disorder. And let me backtrack. A genetic disorder just means there's a gene mutation, right? So it could be 3 or 4 letters that are different. An A could be substitute for a C. So that means it's just a small little change in the A's, T's, C's, or G's. So X-linked recessive now, if you notice on this pedigree, it's a little different. The males, again, are squares, and you notice that the males mostly, if not almost always, have it. And in this case, this pedigree shows the females that are carriers. So you may see that, you may not. And so the males get it more often because, remember, they only have one X chromosome. So they have XY. So if they get the X little r, then they have the trait. Whereas females have to have both X little r's, one from mom and one from dad. It skips generations again because of these carriers who are always females. Males cannot be carriers because they only have one X. Now, if they were XXY, they would be a carrier but still they wouldn't have kids, or they wouldn't be able to have kids. So examples are hemophilia, where your blood doesn't clot, and also color blindness. So another genetic disorder type of inheritance is autosomal dominant. And autosomal dominant, again, you're shaded in if you have it. So that means that people who are autosomal dominant have to have one big R at least. So everybody who's shaded in is big R something, usually big R little r, because big R is lethal. And then the people who aren't shaded in are little r little r. So again, it's autosomal, so you should have equal male-female. It's dominant, so there's no carriers, and so it doesn't skip generations. And whoops, and it's rare to have the disorder and to have big R, big R. So an example of this would be Huntington's disease, where it's a neurological disease that causes spasms, and you get it when you're older. So this is another example of a dominant trait, and that is what's called polydactyly. So if you look at this little girl, then you can see that she has 6 fingers on one hand. And actually, polydactyly is dominant. So people often, and it's usually the case that dominant traits probably are more common. However, this is an example where it's not. If you think about your fingers, right, you mostly have 5 on one hand. Very few people have 6, but having 6 fingers is dominant. So not all dominant traits are more common. Okay, the last one we're going to talk about is X-linked dominance. And so again, what you're going to see here is that you need only one dominant letter. So look at this, would be a male with the trait. So X big RY. And if he had it, like this number 7, and he has this genotype, remember dads give the X to their girls. So every single girl, if he had 10 girls, they would all have this disorder. Now, dads give the Y to their son, which doesn't carry the gene. And so if males have it, then that means their mom has had to have it. But the big pattern to recognize here is that if you have a male with it, then all of the daughters are going to have it. And again, it doesn't skip generations because there's no carriers. An example of this is hypertrichosis, which is werewolf syndrome, and that's just where you grow hair all over your body. All right, and the last thing we're going to talk about is chromosomal disorders. And we talked a little bit about non-disjunction. This is just to remind you, right? And non-disjunction means that the spindle fibers are not breaking correctly. So here, you should have had another one come over here, and so you'd have 2 pairs and 2 pairs. So this would be an example of a gamete with 1 less chromosome, and this would be, again, 1 less, and then these 2 would have 1 more. And so that's what non-disjunction is, and examples we've talked about have been Down syndrome, triple X for females, or any with an extra chromosome. And so here's just a karyotype of non-disjunction happening, and this would be Down syndrome because they have 3 21s. So remember, a chromosomal disorder is where you have an extra or 1 less of a chromosome, or and then a genetic disorder is where you have a genetic mutation. So a couple letters that got deleted, or a letter that got switched, like an A for a G. Okay, and that's it. Have a great day.